showing you a hydrolysis reaction because it's breaking these dimers down into monomers. So enzymes are very specific. We call it the lock and key model because one enzyme is only going to fit with one substrate. When the substrate clicks in with the enzyme, we call it the enzyme-substrate complex. A little reaction occurs, breaks the molecule apart to get the products. This case is a hydrolysis reaction. You could certainly have enzymes that would be doing condensation reactions or other reactions in your body. Why do you need these enzymes? You need them to lower the activation energy, to get these reactions to happen at your body temperature or at your body's pH. One more little part to this story is something that kind of ties in my inorganic compounds, because everything in this picture right now, these are all organic compounds. This is a protein. This, I said, was a saccharide, a sugar. But it is important for you to have inorganic compounds in your body. We talked about water being the most important inorganic compound, but here's another inorganic compound that we need to know, coenzymes. Now, coenzymes is the same thing as saying vitamins. In fact, sometimes bottles, instead of saying multivitamins or vitamin A on them, they'll say coenzyme A. The words mean the same thing. If you stop to think about why you need vitamins in your diet, what they're actually doing, they are actually working with your enzymes in your body to make your body function more efficiently. Co means with. So coenzymes work with your enzymes to help the enzymes work better. It's kind of like WD-40 for your enzyme substrate complex. Coenzymes would sit right in here, and they would help those two pieces to click together better. So remember, the vitamin is not the protein. The enzyme is the protein. The vitamin is an inorganic compound that's helping the two come together. Inorganic compounds are still important. So enzymes lower our activation energy, allowing reactions to happen at our body temperature. And vitamins are not organic. They're inorganic, but they're still important to help your body function well. All right, let's talk about the final organic compound group on your green graphic organizer, and that's nucleic acids. So obviously you've already completed the lab. You've modeled carbohydrates, you've modeled lipids, you've modeled proteins, and you've run organic compound tests for sugar, starch, lipids, and proteins. And you need to know those for your test. You'll notice that you are not going to learn how to model nucleic acids, and you're not going to learn how to test for nucleic acids in this chapter. It's not because we don't know those things and know how to do those things. It's just because we're going to come back to nucleic acids later in the year and spend a good amount of time talking about DNA. And you'll learn how to extract DNA from a substance. You'll learn how to model DNA. For right now, I just kind of want to go over the basics. What's the monomer? What's the polymer? What's the functions? Just so you know some basics about this organic compound group. So nucleic acid monomers are called nucleotides. And there's a picture over here on the right-hand side of the screen that shows you a nucleotide. And it is a little more complex than some of the other ones. It has three major parts. It has a phosphate group, which you see right here. It has a 5-carbon sugar, which you see right here, and a ring-shaped nitrogen base, which you see right here. When you draw this picture in your notes, I would just draw the phosphate group, the sugar, and then I would just draw the base as maybe a square, because you don't need to know that it's a hexagon and a pentagon, and you don't need to worry about that. So you should know that the elements that are within this are phosphate, the same elements that are in sugar, carbon, oxygen, and hydrogen, and then nitrogen. So there's 5 elements here: phosphate, carbon, oxygen, and hydrogen, and then nitrogen. When these nucleotides bond together, they're going to form polymers. There are 3 different polymers. There's actually a lot of polymers that can be formed from nucleic acids, but 3 that I want you to know. So the nucleotide is the monomer, And then there's 3 major polymers I'd like you to know. With each polymer, I'm going to give you a function. So you can separate these on your graphic organizer however you want. The first polymer that can be formed if I do condensation reactions with many nucleotides is DNA. And you've heard of DNA before. So DNA is a polymer of a nucleic acid, and its function is to record instructions for the cell to transmit that genetic information. This is a picture of DNA. You've probably seen this before. It looks like a twisted ladder. And if we zoom in on it, if we take this section and we zoom in here, you look carefully, you'll see the nucleotides. Here's a nucleotide, the phosphate, the sugar, and the nitrogen base. Here's another one, phosphate, sugar, nitrogen base. Here's another one. So you can see how this is just like the carbohydrates. It's just like the proteins. We take a bunch of the nucleotides or the monomers and we link them together through a condensation reaction to form the polymers. 2 other types of nucleic acids that we'll talk about this year that I want you to write down as nucleic acid polymers are RNA and ATP. RNA looks just like that DNA picture right there, and it helps carry out the instructions of DNA. So there's another function of a nucleic acid for you. The final polymer I want you to know is ATP. ATP is a molecule that helps your body get energy, and we'll talk more about that in later chapters as well. But it's formed by nucleotides bonding together. So again, you don't need to know an organic compound test for this. You aren't going to build a model for nucleic acid because we're going to come back to that later. But you still need to know the monomers, the polymers, the functions, the elements, just the basic information about this organic compound group. So looking at your green sheet, you should start to be looking for patterns. You should know the major functions of all the groups. I can cross off this word function and say you need to know the monomers of each of the groups, or the polymers of each of the groups, or the organic compound tests, or the structures. One of the best ways that you can study for this test is to make yourself a blank green graphic organizer. Just draw it out on a sheet of paper and fill it out. I would recommend you do that before you even start studying, because the stuff that you can fill in before you even started studying, you don't need to spend as much time reviewing. Okay? You are going to have to go back and look at your chapter 2 notes. There's about 2 targets from chapter 2. You are going to have to go back and look at the chapter 3 notes that we took at the beginning of the chapter. There's even some prefixes and suffixes in there. You need to know condensation reactions. You need to know hydrolysis reactions. You need to know about enzymes. But the majority of this test and most of the targets have to do with that green sheet. So I'd really recommend drawing it out and filling it out while you're studying, making sure that you're not coming in for that test without actually being able to fill in that chart. And I want you to stop and think about why we build these monomers from polymers. Remember, the idea here is we have 4 simple polymers that can build into countless numbers of molecules for your body. It would be like having one puzzle that you could take apart and put back together over and over again into every different puzzle you could imagine. Different pictures, different shapes, different sizes. It gives your body an advantage. It doesn't have to have hundreds and hundreds and hundreds of compounds in it. It only has to have a few basic pieces, a few basic monomers that it can build up into the different polymers that it needs. Now this chapter is going to be challenging to study for because there is a lot of information to put together. So you're going to want to think about ways that you can study best. Like I said, I would highly suggest making the blank graphic organizer and filling it in for yourself. I would go back over your notes in your outline quiz. I would study from your lab and your targets. But there are some resources at the end of the chapter of your book, and this is one of them. So you don't need to start copying this down right now, but you can make yourself a big note to go into the end of chapter 3 and use this picture. This picture is at the end of your chapter 3 to study. The answer key for this is in the back of your book. So this is just another thing that you can use to review. And this is stuff on the green sheet, guys, the functions and the examples. It also has all of the diagrams, all the pictures that you can study from, and labeling the monomers. This is a great extra review. So people that want just something a little extra, here you go. Use this. It's at the end of chapter 3. Make sure that you filled out that target sheet thoroughly and that you are ready for that test, because there are a lot of details. This is one chapter where you may even want to think about making flashcards. You've done enough studying this year. You've had several tests in this class. You should have a very clear picture of what you need to do to be successful on this test.